Hello everyone. So welcome in this lockdown lecture series of fundamental of plant pathology, and today we will discuss about the role of enzyme, toxins and growth regulator in the plant disease development. So pathogen versus plant, yeah, plant versus pathogen. Plants is always surrounded by the so many disease causing agents. So earlier we discussed abiotic, mesobiotic and biotic. So under abiotic like temperature, humidity, soil condition, similar, and in mesobiotic virus, viroids, and in the biotic fungi, bacteria, nematode, phanerogametic plant parasite, phloem inhibiting, xylem inhibiting bacteria, mycoplasma, phytoplasma. So for that, in nature, generally plants and pathogen are fighting each other. Means one battle is there. Yeah, they always fighting for survival. So plant make some structural and chemical changes to overcome the pathogen effect, and simultaneously pathogen make some activities like secretion of enzyme or toxins to cause a disease in plant. So that one we will discuss in another topic, that is the defense mechanism in plants. So for invading a pathogen to cause disease in plant, it should be invade the host surface, yeah, that host cell. So these are the parts of a host cells. These first is the cuticle, then epidermis, then wax layers, then cutin, cellulose, then pectin layers, then and after that it enter into the cell. So plasma membrane and cytoplasm. So functionally, cell wall is divided into three regions, that is middle lamina, primary cell wall and secondary cell wall. So primary cell wall is made up of a cellulose and pectic substance, and middle lamina, that is called as a cement of cell wall, that is made of a pectin substances, and secondary cell wall is entirely made up by the cellulose. So all you know, the major part of primary cell is the 90% is made up of a polysaccharides like cellulose, hemicellulose, yeah, pectin polysaccharides. And these monosaccharides are the monomer of glucose, fructose, ramenose, galactose, and remaining that 10 percentage it may be proteins or that substances. So for causing a disease, pathogen first land on the plant surface. It may be root surface or it may be leaf surface. So in aerial part, it contact with the leaf surface, and it is covered by a cuticle, or after that is a cellulose. But in root, there is absent of a cuticle layer. It is consist of a cellulose. So after cuticle consists primary is the cutin, because of a cutin is cuticle is made up of a cutin, and after that the lower layer is the pectic layer, then it is made up of a polysaccharide, and after that the epidermis, that is made up of a deposition of a suberin and lignin also, and complete plant tissue disintegration is involved the breakdown of a lignin. After breakdown of a lignin and suberin and all plant layers, pathogen invade in the plant tissues, yeah, plant cells. So what are the mechanical forces created by the pathogen to invade in the further tissues of a plant? So it may secrete the enzymatic, yeah, toxins through which by enzymatic action it can softening of the plant cells, yeah, otherwise it make a aprisorium, make a penetrating peg. These all things are happen in fungi and bacteria. And in case of nematode, it have a needle-like stylet. So through that it can penetrate and enter into the host cell. So after entering, the enzyme produced to make the further penetration easy, and they can secrete the toxins, so they kill the plant's cell and feed on them. So here that picture you can see that is the magnopathy gracie that causing blast disease in rice. So here that GT in the germinating tube. So first pathogen land on the plant surface, after that it germinate through germinating tube, or that germinate tube will convert into a aprisorium. So here you see that the bulbous part, part of that fungus germ tube is the aprisorium. That aprisorium make a slight knob-like structure, that is the penetrating peg, through which it can penetrate or attach to the plant surface, and then it change a hostoria for absorbing a food nutrition. Here you see the nematode that have a stylet, needle-like structure. So through that needle-like structure, it can penetrate the host surface. So how pathogen affect plants? Pathogen, what are the mechanism? What are the activities which throw plant pathogen attack on a host, or what are the activities happen through this that we can weak the plant? So first, the utilizing host cell means pathogen utilize the host cell content by killing host or by interfering with its metabolic process through their enzyme and toxin. That normal respiration, transpiration, translocation of a food, translocation of a water, it can inhibit it through secretion of a toxin and enzymes. So today we will discuss about the enzyme and toxin, means second point जो है that we will discuss in further slides. And by weakening of the host due to continuous loss of a nutrient, because pathogen feeds on host, so it may weak the pathogen, and by interfering with the translocation of a food and minerals of the plant, so proper it inhibit the photosynthesis and other activities, so plant get diseased by the pathogen. So what are the chemical weapons of a pathogen to cause a disease? That is the enzyme, toxins, growth regulator, and polysaccharides. So polysaccharides mainly plugging substances that can hinder or inhibit the translocation of a water from root to other part of a plant. So all plant pathogen except virus and viroid, because virus and viroids always depend on a host machineries, so they cannot secrete any type of a enzyme, growth regulator, toxins. They other pathogen like fungi, bacteria are secreting the enzyme and toxins or growth regulator. So in general, plant pathogenic enzyme disintegrate the structural components of the host cell and break inert food substances in the cell, or affect that components of the membranes, and the and after that they attack on a protoplast of the host cell. So they interfering with its function system. But the toxin, they never attack on the cell wall, otherwise that structural component of the host, they always directly attack on a protoplasm of the host cell and interfere with the permeability of the membrane. So through this they can affect the normal functioning of the plants. And growth regulator, they are actually hormones, so affect on the cell and either increasing or decreasing the ability of a cell division. So also we discussed in earlier letter, that is the hypertrophy, hyperplasia, atrophy, dwarfing, like that. So increasing the cell size, that is hypertrophy, and if hyperplasia, increasing cell number, increasing number of division and hypoplasia due to decreasing cell division number. If tissues development is stopped, then it is the hypoplasia. And polysaccharides seem to play a role only in the vascular diseases, in which they interfere the passive translocation of a water. So next one more things is there. Some that two terms are there. One is the pathogenicity factor and another is the virulence factor. So already we discussed what is the pathogenesis, what is pathogenicity, and what is the virulence. So pathogenesis means development of disease, that events occurs during the disease development is the pathogenesis, and pathogenicity is the ability of a pathogen to cause disease, and virulence is the successful expression of a pathogenicity. Otherwise we can say the measurement of a pathogenicity is the virulence. So substances or compound produced by the pathogen during plant pathogen interaction, which are necessary for a pathogen to infect and cause disease, is or that are the pathogenicity factor. For example, cell wall degrading enzyme, pectin, lyses, otherwise that polygalactonase or some toxins. And virulence factor, that substances or compounds that produced by a pathogen during the pathogen interaction and that are help but not essential for to cause a disease. So most of the toxins and some enzymes are playing role as a virulence factor. So now come to the original topic is the enzyme in plant disease. So enzymes are the large protein molecules that catalyze all the interrelated reaction in the living cells. So on the basis of production, it may be constitutive or it may be induced. Constitutive enzyme, those are enzymes are present always in the cells, and induced, many are produced only when they are required, then when they are needed. So need based production of enzyme is there, then it is called as a induced enzyme. So earlier if we see the work started on a role of an enzyme in disease development, that is the Anton D. Berry, and he reported the role of enzyme and toxin in a tissue disintegration caused by sclerotonia sclerosing. And he give two terms. One is the messerting, means cell separation, and he told that is the due to extracellular enzyme. And second is the killing factor, that is due to the toxin oxalic acid. Earlier he told about oxalic acid is the responsible for killing factor, and messerting means separation of a cell is due to extracellular enzyme. And one more scientist is the Jones. He reported pectolytic enzyme by soft rot bacteria and like that. So what are the enzymes involved in the plant pathogenesis? These are the first is cutinase, cellulase, hemicellulase, lipase, proteinase, pectinase, lignase. These are the enzymes that involved in pathogenesis or development of a disease. So cutinase, cellulase and pectinase and lignase are secreted by the pathogenic organism, at fungi, nematode, bacteria are the well known to produce these enzymes. So enzyme in specific pathogen host combination also we discuss, and virus and viroid are generally not considered as a secreted enzyme. Means virus viroid, they never produce any toxic or enzymes in during the disease development. So enzyme degrade cuticle. First layer of the plant surface is the cuticle. So in cuticle of a many aerial plant parts, it is made up of a cutin, and most of the penetrate wax through mechanical action, but some fungi like Puccinia hordi and Pestalosia melicola are produce a cutin degrading enzymes. So enzyme degrade cutin. So cutin is the main component of cuticle, and many fungi and bacteria produce cutinase degrading enzyme. And fungi constantly produce cutinase in small amount. Some in the constant with the related. Always they produce a small amount of a cutinase enzyme. So because plant surface are coated with thick or thin layer of a cuticle for protection of a prevention of water losses, and it also protect against the pathogens. So cuticle already I told that is a simply made up of a cutin and structural polymer composed of a ester of a hydroxy and hydroxy fatty acid. That is the cutinase are a group of a enzyme है ना, that is hydrolase the plant cuticle, and biochemically cutinase enzyme belong to the serine hydrolases family. Also that cutin enzyme is very essential for the direct penetration of the host cuticle by pathogenic fungi. So one more thing, the presence of a glucose suppress the expression of a cutinase enzyme, cutinase gene, that cutinase protein that is responsible for production of a cutinase enzyme. So enzyme that reaches at higher concentration cutinase are present in the penetration point of the hypha at the infection phase of a episorium. Here you can see that is the plant surface, and after landing of a pathogen it secrete a cutinase enzyme that further trigger and induce the to produce a more cutinase enzyme for softening of a plant surface, and further it invade in a deep seated tissue. So there is a good correlation of a cutinase level and virulence, means pathogen produce more cutinase enzyme are more virulent, they have easy more aggressive. And so cutinase deficient mutant show decrease virulence also reported, that pathogen that organism having low cutin production capacity are less virulence, and cutin gene change the mode of entry also from wound infection to direct infection, means that pathogen that are produce more cutinase they can penetrate directly, otherwise they can penetrate through wound या फिर natural opening like stomata, lenticels, that is the. So pathogen produce higher amount of a cutinase seem to be a more pathogenic than the other, and cutinase inhibitor or antibiotics if we apply to the plant surface, it can inhibit the pathogen penetration. So some examples are like fusarium, penicillium, that are the rotting fungi in the plants. So next is the pectin degrading enzyme. So middle lamella middle lamella, that intercellular cement, we can say the cement between the two cells is the pectin that bind the two cells together. So pectic polysaccharide has the most complex macromolecules in the nature, and pectic substances are the component of a middle lamina, and they act as a intercellular cementing material. So plant pectin is a polysaccharide component of a large number of a glutaronic acid residue. So for successful entry of a pathogen into the host cell, the pathogen should degrade the cell wall component and get invaded into the tissues or cells. So many pathogenic fungi can produce pectinase enzyme during the process of a infection, and pectin degrading enzyme like pectinase. So pectins are the chiefly involved in a soft rotting fungus. So Here we can see the examples of what are the pectin degrading enzyme, that is the pectin methylesterase, polygalactonase and pectin lysase. So how they work on plant surfaces, and what are the mechanism behind this in during the disease development. Earlier scientist, that Jones, that is the pioneer scientist who he worked on the pectic enzymes. So the pectin methylesterase remove small branches of the pectin chains, and then pectin methylesterase have no effect on the overall chain length. Okay, it break the chains, but do not disturb the overall length of the chain, but they alter the solubility of the pectins and affect the rate of which they can be attacked by the chain splitting pectinase. Then pectinase cleaves the pectic chain and release shorter chain proteins containing one or few molecules of a galactonase. Then some chain are also splitting pectinase called polygalactonases, and split pectic chain by adding a molecule of a water, and breaking means hydrolyzing of the chain, the leakage between two galactonase molecules, and other known as the pectic lysase. The last third one is the pectic lysase, that split the chain by removing a molecule of a water from the leakage, and thereby breaking it and releasing product with a unsaturated double bonds. So this is the mechanism. So it also we can say endopectinase and exopectinase. So endopectinase that break pectin chain at the random sites, and exopectinase only break terminal linkage, है ना. तो endopectinase break anywhere from in between the that pectinase chain, and जबकि exopectinase only break a terminal linkage. ठीक है. Then next one is so pectin degrading produce many diseases. I already told you that particularly soft rotting fungi or bacteria. And one more thing is the pectinase enzyme is secreted by germinating spore, means the pectin is secreted at early stages, so they can easily invade the plant cell wall and help in a penetration of the host, and also that liquefy the substances, so that cell wall become a weak and facilitated to pathogen to penetrate. So weakening of a plant tissue and macerating lead to a inter and intracellular invasion of a tissue by the pathogen. Or for example, if you see that lot of the examples are there, that brown rot bacterium, that Irvinia species, Pseudomonas, that here you see the one more pictures, that is the pears, soft rotting of pears, that is garlic, apples, that potatoes, and that citrus, that all rotting fungi and bacteria secreting the that pectinase enzyme. Now come to the next important enzyme that degrade the cellulose. So all you know, the plant cell are protected with a cellulosic cell wall, and the cellulose content of a tissue varies from about 12% to 90%. So 12% in the non-woody tissues of grasses containing 12%, and the about 50% in mature wood plant, that is contain 50% cellulose tissue, or more than 90% in cotton fiber, means cotton fiber is containing more than 90% cellulose, and about 20% in the meristemic parts, whatever the young growing parts, that they contain less or lower cellulose in their content. So Cellulose is the polysaccharide consists of a chain of a glucose molecules, and cellulase is the degrade cellulose. So what are the mechanism and what are the enzymes involved to degrade the cellulose? These are the enzyme. So if we see the enzymatic breakdown of a cellulose result in the final production of a glucose molecules here. So the glucose is produced by a series of a enzymatic reaction. Here you can see that diagrams of that series that carried out by the several cellulase and other enzymes. So C1, one cellulase attack on a native cellulase by cleaving the cross linkage between the chain, and it convert into a modified without cross linkage cellulose. And the second C2 also attack on a native cellulase and break into a shorter chain, small chains, and these are then attacked by a third group of a cellulose, that is the CX, which degrade them to the disaccharide cellobiose, and finally cellobiose is attacked by a beta glucosidase, and that degrade by the convert into a glucose, and that degraded glucose is a soluble sugar, and pathogen can utilize as a food that glucose. So cellulase produced by several phytopathogenic fungi, bacteria, nematode and parasitic higher plants, and softening and disintegration of a cell wall. So liberating a large molecule of a transpiration in stream of a water translocation, which interfere with a normal movement of a water, that is mostly in the wilt, causing fungi and bacteria. They inhibit the translocation of a water. So for example, that cone stall rot, fusarium, Rhizoctonia solani, Fusarium moniliforme, Sclerosium rolfsi. These are the fungus that produce cellulase to degrade the cellulose. Next is the hemicellulase. So it is also a complex mixture of a polysaccharide polymers and major constitute of a secondary cell wall. So hemicellulase polymer include xyloglucan, glucomanone, galactomanase, and hemicellulose degrade the hemicellulose depending on the monomer release. After degradation, they are called like xylans, glucans, galactanase, arabinose, या manase. So that fungal pathogen produce enzymes and oxidative agents such as the activated oxygen and other radicals that also degrade the hemicellulase. That are the sclerosium species that release the reactive oxygens. Next is the lignin degrading enzyme. So lignin is the most complex material, and it always we can say it found in almost in the dead plant material, whatever that is translocating tissues like xylem. So only a small group of a microorganism are capable to degrade the lignin because it is the more resistant to enzymatic degradation. So ligninase is the degrade the lignin. So most of the brown rotting fungus that produce lignin means fungi belong to the basidiomycetes of the class. Hymenomycetes are capable to produce a ligninase enzyme to degrade lignins. So and they characteristically called a brown rods. So ligninase activity is the reason for wood decaying of a fungal infection. मतलब mostly जो wood decaying wood decay through ligninase enzyme secreted by the fungus of Basidiomycetes. One more thing is there, even fungi can degrade the lignin, but they cannot use as a food food source. Yeah, they cannot be used as a food. But some white rotting fungi is there, so they can produce ligninase enzyme and degrade and utilize as a food source. That are the white rotting fungi. So up to this pathogen penetrated the all layer of the cell walls and finally get permission, get means it is inside the host cell. So after entering into the host cell, it have to secrete a enzyme to degrade protein, starch and lipids. So what are the enzymes that responsible for degrading the protein, starch and lipids after entering into the host cell? So first is the protein degrading enzyme. So as you know, the proteins have diverse role in the cellular reaction as enzymes or as a structural component like membrane, that plasma membrane and cell wall also. So in order to establish the pathogenesis, the pathogen are required to degrade many protein of the host origin in the cell. So and almost all pathogenic fungi and bacteria can produce many proteinase enzyme for to degrade the proteins. So proteolytic enzyme produced by most of the pathogen can affect the organization and function of a host cell. So for example, Pyracularia oryzae, Phytophthora infestans, yeah, Helminthosporium oryzae, they can secrete a protein degrading enzyme, that is protease. Next is the starch. So starch is the main reserve of a polysaccharide and found in the plant cells, and starch is the glucose polymers containing two forms, amylose and amylopectin. So most pathogens utilize starch as a food. That earlier we also seen that a lot of fungus utilizing glucose as a food source after degrading a cellulose. So degradation by enzyme is the amylase, and product is the glucose again, and it is used by the pathogen directly. So for example, many Aspergillus species are secreting amylase, which can degrade the starch components. For next is the last, we can say, lipid. So many types of lipids are present in the host cells in form of oil and fats, especially in seed, because lipid is the main source of to conserve energy, storage energy compounds in the seeds. So wax lipid is the most aerial epidermal cells. We already discussed, is the first layer of the plant surface, and phospholipids and glycolipids along with the proteins in the cell membrane, that after decaying of a cell wall, next membrane is the cell membrane, and then pathogen enter into the cells. So lipolytic enzyme in several fungi, bacteria and nematodes, that is lipase and phospholipase, are responsible for to degrade the phospholipids and glycolipids, and in the form of a these are the basic unit of a lipids. So lipolytic enzyme hydrolyze the fatty acid and liberated fatty and liberate the fatty acids that is utilized by a pathogen, and sometimes fatty acids are hyperoxidated by lipogenase or active oxygen molecules. So again that trigger the development of a defense mechanism in plants. So these are the examples of lipid degrading fungi, that is Sclerotium rolopsi, Botrytis cinerea. So now come to the summary of what are the biomolecules present in host cells, and what are the degrading enzymes, and what are the examples of that. So first we can see the cuticular wax, that is the wax degrading enzyme, and example is the most famous example and many times asked in exams, that is Puccinia horridi. And next is the cuticle, that is made up of a cutin, and degrading enzyme is the cutinase, and examples are Fusarium, Penicillium, these are, and pectic. Third is the pectic substances, so it is degraded by the pectinase or pectolytic enzyme, or pectin substances is the major part of a middle lamina, or it is also called as a cementing agent. So and next is cellulose. Cellulose is degraded by cellulase and convert into a glucose monomers. That fungi are the Rhizoctonia solani, Fusarium moniliforme, Sclerotium rolopsi. Or cellulose is degraded by so many series of enzyme, that is cellulase C1, C2, CX, cellobiose, like that, and finally convert into a glucose monomer. And fifth one is the hemicellulose, that is degraded by hemicellulase. And sixth one is the lignin, that is degraded by the ligninase enzymes. And after entering into host cell or in contact to host plasma membrane, they secrete protein, starch and lipid. So protein degrading enzyme are protease, starch degrading enzyme are amylase and secreted by Aspergillus species, and lipid degrading enzyme is the lipase. And one or two more for DNA and RNA degrading also enzyme is found, that is the DNase, deoxyribonuclease, and RNase, that is the ribonuclease enzymes. So now come to the next topic, is the toxin. So what is toxin? What are the basic characters of toxins, we can say. So toxin are non-enzymatic product of a microorganism or a microorganism host interaction, and which is harmful to plant at a low concentration. So they are a low molecular weight compound, mobile in plant, they can move systematically from one place to another place in plant, and do not attack on a structural integrity of a host like cell wall. Earlier I also told that toxin never attack on a structural integrity of a host part like cell wall. They always attack on a protoplasm. So they act by inhibiting what are the mechanism behind? They act respiratory enzymes or other enzymes. They interfere with the vascular movement of a water and nutrient. So if we see the history of a toxins from where it is started, so discovery of a endotoxin of Clostridium tetani or Clostridium botulism, that is Dibbabandi, and Corynebacterium also. They established the role of a toxin in human disease. But after that, some significance of a toxin in plant disease development, that people thinking that maybe these toxins are play a role in the pathogenesis in disease pathogenesis in plants, means disease development they realized. So in 1954, Ernest Guman had asserted that the microorganism cannot be pathogenic unless it is a toxic, unless it is a secrete a toxin. Means microorganism are harmful or cause disease only when they secrete toxins. Okay, otherwise they are not a harmful to plants. So microorganisms are cause disease or damage cell only when they produce toxin or microbial poison, any microbial poison. So in plant pathology, term toxin is used for product of a pathogen, yeah, its host or pathogen interaction, even at a very low concentration, and directly act on a living host protoplast to influence or causes the disease development or symptom expression. So these are the toxin. And one more thing is that the toxin are lethal only microorganism, plant, animals. However, antibiotics are toxic only to microorganism like fungi, bacteria and MLOs. Means toxin are only toxic to plant and animals, not toxic to fungi, bacteria and mycoplasma. But antibiotics are toxic to microorganism like fungi, bacteria and MLOs. So this is the difference between the toxin and antibiotics. So what is the antibiotics? You know definition of antibiotics. So the antibiotics are the substances produced by a one microorganism which is very dilute concentration and have a capacity to inhibit the growth of a other microorganism, not plant or animals. So that's why antibiotics are not a toxins. So in viral infection usually not included in a toxic effect, because viruses do not directly affect the protoplasm of the host. Yeah, it do not affect the functioning of a protoplast of host. Similarly, enzyme causing tissue disintegration are not toxic because they do not affect the protoplasm and only disintegrate the cell wall. Means enzyme those affecting cell wall synthesis or cell wall disintegration are not considered under toxins. Matlab those enzymes that affect protoplasm of the host plasm, host plant, those are the considered under toxins. So finally, the toxin hypothesis experimentally proved with the study of a metabolite of a Cochleobolus, that is earlier is the Helminthosporium Victoria fungus which causes Victoria blight of a oat. In vivo toxins we will discuss in later. So what are the target sites of a toxin in plant cells? These are plasma membrane, peroxisome, Golgi apparatus, maybe chloroplast, mitochondria, endoplasmic reticulum. It may be affect the work of a nucleus, nucleolus, ribosome. These are the target sites of a where the pathogen attack or affect the plant functioning. So what are the effect of a toxin on a host? So there is a three major effects. One is the change in cell membrane permeability, and second is the disruption of a normal metabolic process, and third one is the interfere with the growth regulatory system. So in first sense in cell membrane permeability. So because toxin kill plant cell by altering the permeability of a plasma membrane, thus permitting loss of a water and electrolytes, and also unrestricted entry of a substances including toxins. So and also it affect the cellular transport system, especially hydrogen and potassium ion exchange at the cell membrane. And the second one is the disruption of a normal metabolic process. So it increase the respiration due to disturb the salt balance. Also malfunctioning of a enzyme system like pyrrolidine inhibiting the polyphenol oxidase, and another is the uncoupling of a oxidative phosphorylation. So and third is the interfere with the growth regulatory system of a host plant like auxins, gibberellin. They imbalance the growth regulator ratios in a plant cells, and it may cause hypertrophy, hyperplasia, atrophy like these. So now come to the classification of a toxin. What types of toxins are present in host or pathogen or host pathogen interaction. So earlier toxin are designated as a that phytotoxin, vivotoxin and pathotoxin, but now term that commonly used as a host specific toxin and non-host specific toxins. So according to Wheeler and Luque in 1963, he classified under that the phytotoxins, vivotoxins and pathotoxins. So now we discuss about the phytotoxins. What are the toxins included in the phytotoxins, and what are the examples of phytotoxins? So any product produced by a microorganism that is toxic to plant is called phytotoxins. It is a non-specific in nature, means it affects the lot of plants. It is not specific to particular plant. It produces little or none of the symptom. It is means it is not responsible for to cause a majority of the symptom, and there is no association between the toxin production and pathogenicity. Earlier we discussed the pathogenicity and virulence. So it is not responsible for pathogen, it is not necessary to cause the disease. For example, cochleobolin and alternaric acid. Next one is the vivotoxin. So that vivotoxin term is coined by Diamond and Wagener in 1953, and the compound produced in the infected host by the pathogen and or its host which play a role in the development of a disease, but is not itself the initial initiating agent of the disease. Means it is the vivotoxin cannot initiate the disease, they can enhance the disease. After disease development, they enhance the disease, but they cannot be initiate the disease. So in India, if you recall the history, that is the first or second lecture, so that is Sadasivan in 1955, that he also established a school of plant pathology in Madras, that is Chennai, and he developed a concept of a vivotoxin in India and reported the production of a fusaric acid by Fusarium vaccinificum that causes wilt in the cotton. And this is the non-specific in nature. It produces the majority of a symptom, means it enhances the disease, but it cannot initiate the disease. It enhances the virulence and symptom. Okay. And according to Prigel and Safer, it is a host non-selective toxin. And example are the fusaric acid, lycomycin, and pyrifluorine. Next toxin is the pathotoxin. That is the important toxin, and terms coined by the Wheeler and Luque in 1963. So pathotoxin play causal role in a disease production and produce symptom characteristic of the disease in a susceptible plant. Means it can initiate the disease and enhance the disease. Means that is the main responsible toxin for causing a disease. It is specific in nature, and some toxins are also non-specific in nature and produce characteristic symptom of a disease still in the absence of a pathogen. And they are primarily determine the pathogenicity also. And according to Prigel and Safer, it is a host selective toxin, and example is the Victorian. Other example of a pathotoxin are selective and non-selective both. So in selective, that is T toxin, SC toxin, SS toxin, phytoalternate, PC toxin, and non-selective, that is the TEN toxin, TAP toxin, and faciliotoxin, and that is the respective organism that is produce these enzyme are Alternaria tennis, and TAP toxin is produced by Pseudomonas tabaci, that is wild phytotoxins. So now come to the another classification based on the specificity of a toxin. So toxin in plant parasite interaction we can classify under general toxin, non-host specific toxin, or we can say non-host selective toxin, and another is one is the specific toxin, host specific toxin or host selective toxins. So selective and non-selective term coined by Pringle and Safer in 1964. So non-selective toxins, these are the metabolite product of the pathogen, but do not have a host specificity and affect the protoplasm of many unrelated plant species that are normally not infected by the pathogens. And the specific selective toxins are, these are the metabolic product of the pathogen which are selectively toxic only to the susceptible host of the pathogens. In examples, if we see the pathotoxin, vivotoxin, and phytotoxin like TAP toxin, TEN toxin, faciliotoxin, fusaric acid, lycomycin, pyrifluorine, alternaric acid, coletotin, cochleobolin, these all are the non-host specific toxin, and specific toxin are the Victorian, T toxin, SC toxin, and AM toxin. So now come to the non-host specific toxin or non-host selective toxin. So they affect a wide range of a host plant, produce all or a part of a disease symptom on the host as well as the non-host plant. They increase the extent of a disease. They means they enhance the disease, not initiate the disease. So enhance virulence and symptom development in host, and do not determine the host range. For example, already we discussed, that is the vivotoxin, TAP toxin, TEN toxin, faciliotoxin, pyrifluorine, like that. So one by one, first we will discuss about that TAP toxin. So TAP toxin is produced by the bacterium Pseudomonas syringae, Pathova tabaci, that caused a wildfire disease of a tobacco and causes chlorosis and necrosis in the form of a necrotic spots on the leaves surrounded by a yellow halo. And sterile culture filtrate of the organism as well as purified toxin produce the symptoms identical to the characteristic of a wildfire of a tobacco disease. Not only on a tobacco, but it also affect a large number of a host species that is belonging to a many different families. So strain of a Pseudomonas syringae pathovar tabaci sometimes produce a mutant and that have lost the ability to produce the toxin symptoms. So now come to the structure and mode of action of a TAB toxin. So TAB toxin is the dipeptide compound of the common amino acid threonine and TAB toxinin with the molecular weight of a 260 Dalton. And TAB toxin as such is not toxic, but in the cell it become a hydrolyzed and release TAB toxinin. This is the active toxin and toxic to the plant, and it inhibits the glutamine synthetase that catalyze the synthesis of a glutamine from glutamic acid and ammonia. So due to inhibition of a glutamine synthetase, that means due to inhibition of a glutamine synthetase, glutamine level fall and ammonia accumulation to toxic level, means increasing of a accumulation of a ammonia into the host cell. That ammonia uncouple the photosynthesis and photorespiration and damage the thylakoid membrane also of the chloroplast and result that develop a chlorosis and necrosis of the host cell. So here you can see in the diagrammatic representation that all threonine, TAB toxin that are inactive TAB toxin that degrade in the through aminopeptide in TAB toxin, that TAB toxin inhibit the glutamine synthetase, and due to this the increasing level of a glutamine which accumulate the ammonia, and that leads to development of a chlorosis. Next is the fasciolotoxin. So fasciolotoxin is produced by the bacterium Pseudomonas syringae pathovar fasciolicola and causes the hollow blight of a bean and some other legumes plant, and the localized and systemic chlorosis symptom produced in infected plant are identical to the those product of a plant treated with the toxin also. So what are the structure and mode of action of these toxins is the fasciolotoxin is a modified ornithine alanine arginine tripeptide carrying a phosphosulfonyl group. So soon after the tripeptide is secreted by the bacterium into the plant, and plant enzyme cleaves that peptide bonds and releases alanine, arginine and sulfoalanine. So that phosphosulfoalanine is the functional fasciolotoxin, active toxin. So inhibit the enzyme ornithine carbonyl transferase that cause accumulation of a ornithine and depletion of a citrulline and arginine. So toxin also inhibit pyrimidine nucleotide synthesis and reduce activity of a ribosome and interfere with the lipid synthesis, and also alter the membrane permeability and accumulation of a starch in chloroplast So infected plant and plant treated with the purified toxin also show the symptom like reduced growth. So toxin play a major role in the virulence of a pathogen by interfering with the breaking the disease resistance of the host towards not only the hollow blight bacterium but also several other fungal, bacterial and viral pathogen also. Next is the ten toxin. So ten toxin is produced by the fungus Alternaria alternata, and previously it is called as a Alternaria tennessee, and which causes spots and chlorosis in the plant of many species. And if we see the structure and mode of action of a ten toxin, then ten toxin is a cyclic tetrapeptide that bind to inactivate a protein, that chloroplast coupling factor involving energy transfer into a chloroplast, and it also inhibit the chloroplast RNA polymerase. So the toxin inhibit the light dependent photophosphorylation of a conversion of a ADP to ATP also, and in sensitive species ten toxin interfere with the normal chloroplast development, and it result in chlorosis by disturbing a chlorophyll synthesis. And additionally, it unrelated effect of a ten toxin on a sensitive plants is that means it inhibit the activity of a polyphenol oxidase enzyme that involved in a several resistance mechanism of a plant. So both effect of the toxin, namely stressing the host plant with event that leads to chlorosis, and suppressing a host resistance mechanism also, so that enhance the virulence of a pathogen. It means it causes the chlorosis, and it also suppress the resistance mechanism of a plant. So next is the cercosporin. So cercosporin is produced by the fungus Cercospora and by several other fungus, and it is a unique photoactivated toxin. Means it causes damage, leaf spot and blight disease of many crop plants such as Cercospora leaf spot of a maize and grey leaf spot of a corn, like that. So if we see the structure and mechanism, so it is a perialanine guan, and light is the most critical factor for activation of a toxin, because the light and in presence of a light it become a toxic to plant, and by generating activated species of a oxygen, particularly single oxygen. So initially it absorb light of a 400 to 600 nm and convert into a active cercosporin toxin. Means in direct condition it is a inactive or it is non-toxic. So after absorbing a light, it excited and template stage, and that oxygen radicals oxygen break into two radicals, active oxygen, that singlet oxygen it break the plant cell and by damaging the cell membrane, and causes the ultimately the death of a cell. So next is the lycomarasmine, that is produced by a Fusarium oxysporum causing tomato wilt, and first toxin isolated pure from culture filtrate of the fungus. It is the first toxin which is isolated in pure form through fungus culture filtrate, or it is non-specific, and toxic is a In structure we see that is a dipeptide, and the structure is analogous of a yeast, that yeast growth factor, that is the streptogenin. But in place of a glutamic acid in streptogenin, lycomycin contain aspartic acid, means that streptogenin containing glutamine acid in yeast, but in lycomycin it contain a aspartic acid in place of a glutamic acid. So lycomycin, what are the mechanism? If you see the, because lycomycin chelate the iron and dislodge loads from the essential sites, and also damage the permeability of a leaf cell, means it's make unavailability of a iron for a plants, or because pathogen making a chelating, that is a ring of a iron, so plant showing that symptoms like iron deficiency. So fusaric acid, if that produced by a fusarium oxysporum forma species vasinfectum and causing wilt in cotton, and produced by a fusarium oxysporum forma species vasinfectum already. So chemically it is a 5N-butynyl pyridine 2 carboxylic acid, and damage as like lycomycin, that plasma membrane, and causes leakage of a ion, and it also increases the rate of a respiration and inhibit polyphenol oxidase activity, and also chelate the irons and damage membrane permeability by a leaf cells. Some other non-host specific toxins are the like fumaric acid, that is produced by rhizopus, oxalic acid produced by sclerotium, alternaric acid, that is produced by many alternaria species, sarcotolonium, that is produced by Ophistoma ulmi, that is the causing agent of dust elm disease, that is means forest pathogen, and fusicocin, that is produced by Fusicocum amidoli, and ophiobolins, pyricolarine, fusaric, and lycomycin acid, that already we discussed. And now come to the host specific toxin, that host selective toxins. So they act on the specific host of a pathogen and produce all the symptom of a disease, and also toxin determine the host range and specificity of the host pathogenicity factor, and examples are Victorine, acetoxin, and T toxins. So Victorine or acetoxin, how it is produced by a fungus Cochleobolus victoria, earlier it is called Helminthosporium victoria, which causes Victoria blight of a oat, and this fungus appeared in the 1945 after the introduction of a widespread use of a oat variety, that is Victoria, and it derivatives, so all of which contain the VB gene for a resistance to a crown rust disease, means that Victoria variety of a oat containing a VB gene, that is resistance for crown rust disease in oat. So after that Cochleobolus victoria infect the basal portion of a susceptible oat plant and produce a toxin that is carried to the leaves and causes a leaf blight and destroy the entire plant. So Littensberger in 1949 demonstrated the culture filtrate of a fungus that contain a toxin that could be responsible for disease. After that Wheeler and Luque in 1954 they gave the name Victorine. Earlier it is a pathotoxin, but now it is a host selective toxin. So if we see the chemical structure and mode of action, so Victorin is a complex chlorinated partially cyclic pentapeptide with molecular weight of a 840 Dalton, and the primary target of a toxin seems to be the cell membrane, and it bind to several membrane proteins and some yet unknown mechanism, and alter the metabolism of the host. So leakage of electrolytes from membrane, it may detected within the 2 minutes of toxin treatment to the host, and initiate also hypersensitive responses, which leads to programmed cell death of host, and induce senescence in host plant, and also it play a dual role, means it also act as a elicitor. So what do means elicitor? So elicitor term is given by knee, and it is a pathogen derivative molecule that trigger the defense response, resulting in improved resistance to the pathogens. So next is the SC toxin. So it produced by Cochleobolus carbonum races first, and isolates is a tox which causes northern lip spot and ear rot in a maize, and anamorph of Cochleobolus is the bipolaris, the Helminthosporium giacola. It toxic only on a specific maize line, that's why it is a host specific toxin, and toxin discovered by the Suffer in 1965. If we see the structure and mode of action, so it is a cyclic tetrapeptide with the structure of a cyclo-D-proline, L-alanine, D-alanine, L-leo, and acetoxin inhibit the enzyme histone decataalase, that reversible deacylate the core histone, that is S3 and S4 of the chromatin. So by this the toxin alter the gene structure and function. So inhibition of a decataalase also affect the expression of a defense gene, and allowing a fungal activity to progress to penetrate the host. So resistance cone lines that they have a gene HM1 coding for the enzyme called SC toxin reductase, that reduce and detoxify the toxin. So we can say the susceptible coron lines, that they are lacking this gene, and therefore cannot be defend against the pathogen, means the resistance coron variety have a gene SM1, but the susceptible coron variety is lacking the gene. So next toxin is the T toxin, it is also called HMT toxin. So it produced by a race T of a Cochleobolus heterophorus and anamorph of a bipolaris medis, and earlier it is also called a Helminthosporium medis, that causes the southern corn lip blight. So first appear in the United States in 1968, and it is spread throughout the corn belt in a 1970. So attacking only corn that had the Texas male sterility TMS cytoplasm. So the ability of a Cochleobolus heterosporus race T to produce T toxin and its virulence to corn with TMS cytoplasm are controlled by one and the same genes. So T toxin does not seem to be necessary for the pathogenicity of a Cochleobolus heterosporus race T, but it increases the virulence of the pathogen. And the T toxin act specifically on a mitochondria of a susceptible cells, which are rendered non-functional and inhibit the ATP synthesis. Also react with the receptor protein molecules UFR13 located on the inner mitochondrial membrane. So next is the Alternaria alternata toxin. So several pathogens of Alternaria alternata attack on a different host plant, and on each they produce one or several multiple form of a related compounds that are toxic only on a particular host plant of a each pathogen. So some of the toxins and the host on which they are produced and affected, that are the AK toxin causing black spot of Japanese peanut, AAL toxin, AF toxin, AM toxin, ACT toxin, ACL toxin. So ACL toxin on rough lemon and SS toxin on a sugar cane. So as an example of a Alternaria alternata toxin, that AM toxin is produced by the apple pathotype of a Alternaria alternata, known previously as a Alternaria mali, and the causes of a Alternaria leaf blotch of a apple. So now come to the what are the differences between the host specific toxin and non-host specific toxin. So if you see the definition, so that host specific toxin, the substances produced by a pathogenic microorganism that at physiological concentration is toxic only to the host of the pathogen, and so little or no toxic against a non-susceptible host. So in case of non-host, it affects several host plant, and it is not specific to a particular host. If we see the pathogenicity and virulence factor, the non-host specific toxin increases pathogenicity, or it act as a pathogenicity factor, means it have a ability to cause disease. But in non-host specific toxin, they are virulence factor, means they enhance the disease. They are not responsible to cause disease, but they enhance the disease, they increases the disease intensity. In symptom cases, we can say in host specific toxin alone produce all the characteristic symptoms, but in non-host cases, these toxins produce all or part of the disease symptom. And host range, they are determined, the host range of the pathogen, means they are specific to host, but in non-specific toxin they do not determine the host range of the pathogen. Type of toxin की बात, they are the pathotoxin of a Wheeler and Luque, and they are the vivotoxin by Diamond and Weger. Example: Victorine acetoxin, T-toxin in under host specific toxin, and TAP toxin, TEN toxin under non-host specific toxin. And one more difference in enzyme and toxin, because we already covered two topics: the role of an enzyme in plant disease development and role of a toxin in plant disease development. So enzyme are generally large protein molecules that catalyze organic reactions, and toxin are the low molecule weight and non-enzymatic factor and produced by many plant pathogenic fungi or bacteria. If you see the mode of action, then they act on a structural component, structural part of a plant, that is cell wall, and in toxin case they act directly on a protoplasm and will not act on a structural component of a plant, and if we see the molecular weight, the enzyme molecular weight is always high, and in toxin are very low. So importance in disease development, they are primary factor and they are secondary factor, means mostly they are pathogenetic factor and they are virulent factor. So metabolites की बात, if we see the metabolite, they are primary metabolites and these are toxins are the secondary metabolite, and in specificity they are high. Enzyme are highly specific produced by all pathogen except virus and viral. But toxin, we have seen that specific and non-specific both are available. And killing of a cell, they are killing indirectly because they disturb the cell wall, integrity part of a plant, that is a structural component, but they are directly act on a protoplasm, ectoplast. Chemical nature, they all enzyme are protein, but in case of toxin they are non-enzymatic. Concentration act as a appropriate concentration, and they are extremely toxic at a very low concentration. So example, cutinase, polygalactonase, cellulase, pectinase, tabtoxin and victorin. So now come to the role of a growth regulator in disease development. So as you know, the plant growth is the result of three factors, that is cell division, cell elongation and cell differentiations. So mainly growth regulator are divided in two parts. One is the growth promoting substances and another is the growth inhibiting substances. So auxin, gibberellin, cytoguanine are growth promoting substances, whereas ethylene and abscisic acid, these are the growth inhibiting substances. And that growth regulator basically they imbalance the hormones in plants and cause hypertrophy and hyperplasia in plant tissues, which may leads to stunting of plant, excessive root branching, excessive foliation in plant. So plant growth is controlled by group of naturally occurring compounds with hormonal action, and that are often referred as a plant growth regulator. So major group of plant growth regulator are the auxin, gibberellin, kinetin, ethylene, and the symptom induced by the plant growth regulator are the stunting hai, overgrowth hai, galling, the gall formation, root branching, like that. So now come to the auxin. So auxin are the major growth regulator in plant and are defined as a growth regulator whose major mode of action is the cell elongation, and resemble with indole 3 acetic acid in their activity, and the naturally occurring auxin is the indole 3 acetic acid, and it is continuously produced in a young meristemic tissues of the plant and move rapidly to the older tissues. So if auxin concentration is more, it concentration is reduced by the enzyme indole acetic acid oxidase. So basically pathogen inactivate the indole acetic acid oxidase enzyme. So by inactivating the indole acetic acid oxidase, the auxin production it increases, and in some cases they inhibit the production of a indole acetic acid oxidase enzyme. So by this the production of a auxin is reduced. So in case it may be cause hypertrophy, it may be cause atrophy. So important diseases are these in that is the cone smart clubroot of crucifer, southern bacterial wilt, that is Rollistonia solanacearum, earlier it is Pseudomonas, and crown gall, that is agrobacterium. Next is the gibberellin. So gibberellin, a century ago, the rice farmer in Asia noticed some exceptionally tall seedling growing in their paddy fields. Before these rice seedling could mature, they mature and flower, they grow so tall and spindle shape in top part. So in Japan, the growth pattern become known as a Bakani, foolish seedling disease of a rice. And in 1926, scientist Kurosawa in Japan discovered that the disease was caused by a fungal pathogen, that is the Gibberella fusicori. And by 1930, Japanese scientists had determined that the fungus produced hyper elongation of a rice stem by screening a chemical which was given the name gibberellin. So if we see the gibberellin is first isolated from the Gibberella fusicori, and the best known gibberellin is gibberellic acid in nature, and in the past year scientists have identified more than 80 different gibberellins, and they are occurring naturally in plant, and sparing of a diseased plant with the gibberellin overcome some of the symptoms like stunting, because it increases the plant growth, the elongation of stems, so caused by several viruses or molecules. So pathogen indicating that gibberellin involved in the disease development. So next is ethylene.